So I'm going to talk about sex differences in the brain. And really, why do I care about sex differences? And first, it's simple scientific curiosity. But secondly, and probably more importantly, if you want to understand disorders of mental health, you have to understand the differences in sex proportions and how they might relate to differences in the brains of men and women, males and females, all around. So let's start with a bit of epidemiology. Autism is 4 times as likely in boys as it is in girls. In many ways, it's considered to be the prototypical male brain syndrome. ADHD, attention deficit disorder, is 3 times as likely in boys as in girls. In fact, if I were to ask you to picture a child with ADHD, I'm sure most of you would think of a 9 or 10-year-old boy who can't sit still, who can't pay attention to anything for more than a few seconds at a time. Obsessive-compulsive disorder, a disorder marked by intrusive, repetitive thoughts, is one of the few mental health disorders that's equally prevalent in men and women, or boys and girls. But even there are some striking differences in onset. Onset before puberty is more likely in boys. Onset after puberty is more likely in girls. And that's what we see again and again in these childhood mental health disorders. Early onset disorders such as autism and ADHD are more likely in boys. Later onset disorders are more likely in women and girls. Alzheimer's, you heard Dr. Einstein mention already, is more prevalent in females than males. And same with chronic pain. Chronic pain syndromes are about twice as likely in women as in men. Depression and other anxiety-related disorders are 3 times as likely in women as in men or girls than in boys. And the eating disorders overall are 5 times as likely in women as in men, and some of them range to be as high as 12 times as likely in women as in men. These are striking differences, not just subtle effects, but twice as likely, 3 times as likely, 4 times as likely in one sex than in the other, or one gender than in the other. So if we want to understand mental health disorders, want to understand diseases of the brain, we really have to understand why are they so different between men and women. Now, one of the ways that I go about in studying them is using animal models. Now, one of the main reasons for going to animals, going to mice in my case in particular, is genetics. More and more of these diseases are being recognized as being heritable. So if you have one twin who has the disorder, it's very likely the other twin will have the disorder as well. Unrelated boys and girls are less likely to share the disorder. And we're starting to get more and more information about what are the genes that are involved in the heritable components of these disorders. But knowing that it's a particular gene that's implicated in a particular disease doesn't help us understand the mechanism of how and why that gene is actually involved. One of the things that's a bit surprising on the face of it is that a mouse, a small furry little rodent with a tail, actually shares 92% of its genes with us. So if we identify a gene as being likely implicated in autism or ADHD, there's a 92% chance that that same gene or very similar ortholog of that gene exists in the mouse as well. And if we can find the gene in the mouse, we can begin to change it. We can knock it out or remove the gene, or we can upregulate it or downregulate it, or induce the same mutations that exist in human populations. And we can begin to really understand the mechanism of what does the gene do, and how might the mechanism of how that gene works potentially lead to the disease we're trying to study. But this, and Dr. Einstein alluded to this already, this is where it gets a bit surprising in the literature. Almost all studies consist of male mice only. In fact, for every study in the literature that exists that has female mice in it, there are going to be 5 studies that have used male mice alone. So does it matter? The brains and behaviors of male and female mice differ, just like they do in humans. In fact, you can find anxiety differences in how likely a male mouse is going to go into an open, brightly lit area versus hiding in a shady corner versus a female mouse. And there's differences in the brain, in local cell types and structures, in a few key areas, in particular the bed nucleus of the stria terminalis, the amygdala, and the hypothalamus. Each of these brain areas that have also been implicated in some of the mental health disorders I described earlier. So given that these disorders share some of the, or influence in some of these same areas, and there are sex differences in it, will it make a difference if you choose just to use male mice when you're studying a particular disease or not? And I'm going to give you one example which has been quite striking about where such a difference is really very prominent, and that's from the case of chronic pain. So as you remember from a bit earlier, chronic pain syndromes are twice as likely in women as in men. And there's a series of scientists, including some here at SickKids with Mike Salter's group, as well as Jeff Mogul over at McGill, who have spent years and years beginning to work out the mechanism of how is chronic pain actually signaled in the brain, and what might be, if we understand the mechanism of how chronic pain works, what might we do to be able to relieve it, treat it, whether with pharmacology or with other techniques. And what they have identified over years and years of research is that it's really the immune cell of the brain, the microglia, that are key to mediating chronic pain in the brain. And they've even gone down to some of the more molecular mechanisms of what receptors and what signaling pathways in the microglia are responsible. But all this research was done in male mice. And a few years ago, one of their students, Josie and Mappelbach, began to ask, well, the same thing happened in female mice. And female mice and male mice express the exact same pain behaviors. So you could expect that the mechanism would be the same. Yet it turns out, in their studies as they were looking at it, they did the same manipulations in female mice, and they found that microglia had absolutely nothing to do with pain mediation. Instead, it seems to be, though this is still a bit less uncertain, that it's a different immune cell, the T cells, that are responsible in female mice. Now, this is a bit of an extreme case. You really have two entirely different cell types seemingly mediating the same behavior and the same outcomes that we see. But now imagine that you've just stuck with the male mouse model initially. You found the signaling pathway. You can begin to manipulate it with pharmacology. You can create drugs for it. You can treat it. And even if you did that perfectly, went all the way to the end, it would only be successful in about a third of all the patients you would see in the clinic. So this is really where a classic case of where having to use both males and females will begin to illustrate where the differences arise in some of these diseases and how we can treat them. And if you do it in one sex only, you would be making a mistake. Now, I'll back up for a second. Why would you use a single mouse strain to begin with? And it's really a question of statistical power, ethics, and cost. When we use animal models, we're obliged to use as few animals as we possibly can to effectively answer the question that we're trying to answer. Now, to reduce the number of animals that you can use, you want to limit the variance, the variability between them. So imagine you're trying to look at a gene that's looking at the effects on height. So you're thinking that this particular gene will cause 2 inches of growth. Now, we all know that men are somewhat taller than women on average. So if you were to do the study in men only or in women only, you would have less variance than if you included both men and women in the study, because the height difference of men and women would increase the variance that you see. So that's the argument for using one sex only in that particular case. It reduces the variance, and the same is true in mice. Now, why do most scientists use male mice only? And there, the logic is a bit more tendentious, but effectively, it comes down to the assumption, and it's been proven mostly incorrect, that female mice are more variable than male mice. And usually that goes back to the estrous cycle, or the mouse equivalent of the menstrual cycle, where because hormones cycle in female mice, that causes some differences in behavior depending on what stage in the cycle they're in. Now, that is true, and it does change behaviors, but it's changing it less and less than we thought. And certainly the case of looking at chronic pain is a good example of where studying one sex only might give you more power, but might not answer the question you really want to answer. So different organizations, such as the National Institutes of Health and the Canadian Institutes of Health Research, are starting to mandate that we use both sexes, or at least justify why we're not using it. And it's going to increase the number of animals being used, it's going to increase the cost. But in some cases, it's going to answer key questions. In most cases, the answer will probably be the same. The mechanisms are not that different between male and female mice. But when it's different, we can begin to really understand something about these mental health disorders that really do vary considerably in ratio between males and females, men and women, boys and girls. And we have to understand why they do so. We have to study both sexes to really be able to get at mental health disorders and to understand how the brain works. Thank you.